Neda Jahanshad from UCLA.
Thanks, Jason. I'm here to present multi-site genetic analysis from the Enigma DTI working group with a lot of collaborators from the US, Europe, and also Australia. And by multi-site here, I mean we're doing a meta-analysis for genetic studies. A search for meta-analysis revealed over 400 articles in Nature Genetics in the last 10 years alone, and that's about 20% of our papers. So what exactly is meta-analysis? Meta-analysis, when you look it up in the dictionary, it's a quantitative statistical analysis of several separate but similar experiments or studies in order to test the pooled data for statistical significance. Many meta-analytical studies of genetics have led to important discoveries for traits like neurodegeneration, psychiatric disorders, and behavior and cognition. And we found several genes have been discovered through the use of meta-analysis. Additionally, more recently, the field of imaging genetics has taken off. And in the last year, several features taken from structural images have been used for genetic association studies, including hippocampal volume, intracranial volume, and head circumference. And one study by this guy right here, and many others in the room, was a study of Enigma, and also combined with the charge that they had over 26,000 images. So they were able to do a meta-analysis on that many images and discover variants that do affect brain structure. If you're not familiar with genome-wide association studies, what we're doing is essentially taking a phenotype from the images, in this case it could be intracranial volume, and associating it with every SNP or variant along the entire genome, running an association, and then establishing a significance threshold. And this is these are all the positions along the genome that we're testing. And on the y-axis, we have the negative log 10 of the p-values of association. So the higher the dot falls, the more significant the association is. And here we have a strict threshold for genome-wide significance. Now, white matter is also very interesting because diffusion tensor imaging has been used to find out several aspects of white matter are heritable, and a lot of risk genes for various diseases do link to variations in the white matter. And there are several recent papers that use diffusion tensor imaging to show this. Additionally, genome-wide associations have been done using DTI phenotypes. Most of these are single-site studies of white matter integrity, either using a global measure of FK or a general factor. Their findings have led to their top SNPs being in some candidate pathways, but no genome-wide significant findings have been found in such studies. However, compared to 26,000, these sample sizes have been rather small. So this brings up the need for a multi-site DTI analysis. This can greatly increase the number of subjects, and maybe we can reach some genome-wide significant findings. A few things we have to figure out before we do go ahead and do a multi-site analysis is, would a track from one study be the same as a track from another? And how reliable would a track be if different imaging protocols and different resolutions were used for scanning the images? Now, several DTI reliability studies have been done in the past, but a lot of these focus more on planning for future studies and not exactly for merging data that already exists. And if we want to pool data that already exists, we are able to get more people interested and allow for more groups to join in. So we set up a working group to combine some DTI scans, and some of our initial steps was to first create a template in the same space as everyone's images, and run a widely used track-based spatial statistics from the FSL group, fairly common and can produce to get reliable results. So in our template, we combined data from 400 scans, 100 from each site, 50 males, 50 females from each site, healthy adults. The age range for everyone in this template was between 18 and 85, so we got the entire adult range, or most of it, in there. And the interesting thing here is that all these imaging protocols were fairly different. All of them had a fairly high number of diffusion and acquisition gradients. The voxel sizes were very different, and the field strengths were different. The scanner types were also different. So what we were able to do is use FA maps from all these different images. I said different a lot because they are different. And kind of merge them together and create a minimal deformation template, which we used to register all images to. We additionally had 100 scans from a site that was not included in the template, and we used that for validation purposes. So now, before we get to this GWAS, would different tracts from different populations or data sets even be similarly heritable? So would one tract from one set of images exhibit the same heritability from that same tract in a different data set? And if so, could they be used as endophenotypes for GWAS regardless of the protocol? So let's find out. In this study here, what we did is a multi-site heritability analysis. We had two family-based cohorts, one which Peter works very hard with, and is GOBS, made up of over 800 Mexican-American individuals from extended pedigrees, and also the Q10 sample of European ancestry, and these are all twins. So two different types of family models as well. When we do a heritability analysis on regions of interest from our DTI template, we find, in general, the red, which is the Q10 sample, the twin sample, and the green, which is GOBS, follow a similar trend. Most of the tracts are fairly heritable. This is over 40% heritability. The two exceptions are the fornix and also the cerebrospinal tract, which show a lot of variability or very low heritability in both sets. Now we want to meta-analyze this heritability, and what we do, as Peter mentioned before, is to run meta-analysis two different ways. One weighting by the number of subjects in each heritability, used in each heritability estimate, and also using the standard error of this heritability estimate, which we calculated from solar that Peter was just talking about. We note that these two additional lines, they're both in the middle. It could be kind of hard to see in the back, I apologize, kind of fall right in between the two studies. And again, the fornix and the cerebrospinal tract do not show consistency between the different populations. Now, whether that's because of imaging protocol or differences in ethnicity, it is unclear, but we are removing these from future genetic studies just to reduce our multiple comparison issues. We also ran these analyses on a voxelized level, and these are just some pictures to show you. This is voxelized unfocused bulletin. Additionally, we ran the mega-analysis, which Peter just spoke about, and this was, again, using solar. These two lines on the bottom are the two different types of meta-analysis along each of the regions of interest. And the mega-analysis, you see, we were able to capture more heritability within the entire population of all 1,100 subjects. And these have a slightly tighter confidence intervals, or standard errors. Again, the fornix and cerebrospinal tracts do not really exhibit high heritability. So from this, what we were able to do is a single-site genome-wide association, where we use the average FA in each of these regions of interest as a phenotype for the GWAS. These images just map out the percent heritability. However, we note that at a single site that we used, there was no significant genome-wide association findings, and everything kind of along the norm, or as would be expected. So that wraps up the first project from the Enigma DTI working group, is to kind of establish what is heritable within these FA maps. And now what we are doing is creating a set of protocols for everyone who wants to join in to follow. These include some pre-processing steps, some initial quality control, basically creating the FA maps, a registration to our Enigma template, and also the skeletonization of each of the subjects within your cohort. We have some protocols to do some intermediate QC to look at the projection distance, for example, and make sure everything is in order when you're running TBSS. And once you have everything registered to the template, we have automated scripts that will extract the average FA within each of these regions of interest that we're looking at. And also we have protocols to help you do some final quality control, make sure the ROIs actually do line up with your image, make sure you run some histograms, and make sure you don't have any crazy outliers. After that, we provide protocols to run the association and and any other analysis that you want to do within this protocol, or within this, using this template with this protocol can also be done. Finally, I'd like to thank absolutely everyone in the working group that's been on phone calls to help get this project going, get this initial analysis out. And also, many sites along the way have expressed interest in Enigma DTI and getting it up and running, so I'd like to thank everyone who's expressed interest. It's kind of motivated us to get this project moving along. And we are welcoming anyone else who wants to join the Enigma DTI effort. So if you have diffusion tensor imaging scans and genome-wide data, and you're not able to find significant finding, but you kind of want to join in this effort and do a large meta-analysis, please can sign up at the Enigma website or send me an email, and we will include you in the group. Thank you.
I have a question. So the support, usually when doing support constructions, the heritability for smaller structures is usually lower, mainly due to measurement error. Do you see the same thing?
Yeah, actually the fornix and the cerebrospinal tract are both fairly small structures. Cerebrospinal tract also often gets cut off in some of the DTI images that are noticed. So yeah, I think some of it could just be imaging artifacts, that the heritability is not high, but it agreed.
Is there any lack in epigenetics? Basically, there's 14 replicated alleles across labs We have samples before treatment and no treatment normalizes range. I'd like to get epigenetics done afterwards because I'm not sure we're normalizing the genetic expression, meaning the vulnerability is there, even though we can normalize at a particular time of development. So I'm just curious, epigenetics was mentioned, we're in lab, this is for NIH research, where we could get epigenetics in 14 alleles. Okay.
Dr. Peter.
Just to repeat that question a little bit, so we should have a microphone. She was interested in understanding about epigenetics, which like methylation and things that are not just the genetic code, and Danny lab and stuff like that. Okay, thank you very much, Neda.